Hello everyone. Today we will be talking about antigenic structures and virulence factors of Streptococcus pyogenes. I have already talked about Streptococcus pyogenes in my earlier videos, and if you want to know more about its morphology and characters, you can go back to my video on classification of Streptococcus pyogenes. So here, since we are talking about antigenic factors and virulence factors, so there's a difference between antigenic factors and virulence factors. So first let's again go back to the structure and see whatever is present on this streptococcus. That is M protein, lipotecoic acid. It has a capsule. It has a thick cell wall, thick peptidoglycan cell wall. Then it also has a plasma membrane. It has certain adhesins. That means by which it adhere to the other cells, and it has some certain invasions by which it invade the host tissues, and it has certain exotoxins also. So coming to its antigenic factors, or these are the ones which can evoke immune response in the host cell. So they are responsible for production of antibody from the host cell, and this antibody then binds to the antigenic factors of this particular bacterium. And what will happen? These antigen antibody complexes can be detected by certain tests. So the particular type of strain can be known whether this particular antigen is binding with this particular antibody. So what can be the strain? So this can be diagnosed, can be used in diagnostics, and this way we can diagnose the through the antigen present, we can diagnose whether this particular strain is present or not. So what are the antigenic factors that are present in this particular streptococci? That is hydrolunic acid capsule, cell wall, peptidoglycan, group specific carbohydrate, protein, lipotecoic acid, fimbriae, cytoplasmic membrane, and even pili covered with lipotecoic acid are the antigenic factors which can generate antibody response in the host cell, and they can be used for diagnosis purposes. Now, what are the different toxins and virulence factors present in this particular strain or this particular streptococci? The toxins which are also present are poisonous substances, and they are toxins when present, they are poisonous, and they can be produced by microorganisms, and that can harm cells and tissues of the host. So here we will be talking about streptococci toxins in the next few slides, and the virulence factor which I have represented with VF. And the virulence factors are the mechanisms that pathogen use to cause disease. So moving on to the next slide, these are the different toxins. Let's first talk about toxins. One is hemolysins. As the name suggests, that they lyses the heme part. So streptolysin O is one of the hemolysin, and it's the oxygen labile compound, and it is also heat labile. While streptolysin S, which is soluble in serum, and this is non-antigenic in nature, and this is oxygen stable. So this oxygen stable compound is the one because of which this streptococci can survive in um, surface of the blood agar plates, and it can tolerate oxygen up to a certain level, while streptolysin O is not able to tolerate the presence of oxygen. So when we are growing it on the artificial media, that is on blood agar plates, this is streptolysin S is helping for the growth or colonization of the bacteria. Let's talk more about streptolysin O. This is hemolytic, I have already told you, and it's an exotoxin. Exotoxin is basically a protein in nature, and it's produced by gas, that is group A streptococci. The representative of group A streptococci is Streptococcus pyogenes. So group A streptococci is also called by a short form, that is gas, and this hemolysin O is responsible for characteristics, that is beta hemolysis. Group A streptococci produces beta hemolysis on blood agar plates. So this is the one, and if you want to know more about beta hemolysis and about the classification of streptococci, how it is kept in group A, you can go back to my video that I have put on classification of streptococci. And I will be very happy to receive your comments and your critical view on my presentation. Please let it know through your comments. Let it know to me by putting your comments. So coming back to streptolysin O, it is toxic to human cells and can cause tissue damage. And that is how toxins work. They are causing damage to is a problem. It's not balanced production of T cells. So you can always read about superantigens on my video, which I have put on superantigens. You can go back and find on my site itself. There are certain enzymes which act as virulence factors of streptococcus, and these enzymes are streptokinase, which is fibrinolysin. It lyses fibrinolysin, and deoxyribonucleases, which are also called as streptodornase or DNase, and then NADase, hydrolunidase, and C5a peptidase. Now, how they are working?